And this also includes dose predictions to man. Secondly, when you go into and start to develop your formulation to market, especially if you have, a challenging compound, could be low solubility, or you might want to have a extended release, tailoring the drug release to meet some clinical needs, so you get your drug exposure at the time and amounts when needed for optimal effect. Typically what we do today is very much a trial and error, where we try everything and do perhaps a lot of in vivo studies and then eventually come to a good product. But if we have the right predictive methods, we could target in vivo studies more to be confirmative, and that could reduce both time and of course costs a lot. And in that same, as a consequence of the same thing, we also could reduce the use of animal studies, because that would be, in the lack of good in vitro predictive methods, perhaps a lot of dog studies, primarily pig studies possibly, are done to sort of find the best formulation, and we could reduce that. And there is also a need here to, perhaps sometimes we still need these sort of models, but then to really know when those models are predictive and not. This also play a role when it comes to, manufacturing and setting criteria for quality criteria for manufacturing. Historically, and that's the normal way today, you set these acceptance limits, what is acceptable to put out to patients, based on very conservative historical criteria. Whereas if you have the tools where you really could link the pharmaceutical quality to the exposure in the patient, the effect in the patient, you could start to set meaningful quality criteria, which both could, which could reduce costs while still maintaining quality to patients. And finally, perhaps in a regulatory context, we often have to do in vivo bioequivalence studies. That is, okay, you compare two different formulations, look at plasma concentration profiles, which could be fairly big studies. And these are done to link your perhaps clinical trial formulation to commercial formulation, or if you do changes in your manufacturing after launch, you need to do this study to prove that you still have the same clinical efficacy out of your product. And that could generate a lot of in vivo studies. And it's not only the concern of in vivo exposure and cost of studies, but also if you, let's say you fail a late stage bioequivalence study between your commercial and clinical trial material, then you would delay your NDA probably. So that's not such a good thing. So if we could find out where we could rely in a larger extent than today on predictive methods to do this bridging, we could take away some of the risk and time from the project timelines.
Bertil, could I just make a comment there?
Sure.
When we talk about the expected impact on the R&D process, we actually mean both R&D, research and development. I think Bertil's articulated very well the potential impact on the development process. Real impact here in research as well, and streamlining what we're doing in drug discovery. We've got all sorts of challenges in terms of developing formulations for exploratory toxicology and regulatory toxicology studies, where we're driving exposure way beyond that we'll ever see in human clinical studies. And quite simply, we have very few truly predictive tools for formulation selection for that type of study, which means that we resort to doing a lot of toxicokinetic animal studies very early on, which are time consuming and could be eliminated if we had access to much better predictive tools.
Yeah, good point. So this slide is just to highlight that such a project as this needs to be approached on different levels, from areas where you traditionally think more of as research areas we define where we lack fundamental understanding of the underlying mechanisms. But we cannot just do that. We also need to sort, in some areas it might be that there is a lot of good things out in the literature, but it hasn't been integrated and transformed into methods that make sense based on our understanding. And furthermore, we might have methods available, but the rational use of these methods are not really known. And as I said, often the validation work is more of anecdotal character. So by using industrial data, experience databases, we could sort of find optimal use of these methods. And of course, there is also a feedback loop where we, based on this sort of database analysis, could find the gaps and put up new hypotheses and solve the problems. So the suggestion now here in the material is to organize this as four different work packages, but I would say this is just the first suggestion, and other approaches could for sure be considered. But we divide it into physical-chemical tools where phys-chem properties are related to predictions of exposure and perhaps use of enabling formulations. In vitro tools, including dissolution, permeability, mainly, also intestinal stability. In vivo tools and understanding, then more relating to the physiological aspects, understanding of the relevant physiology, also including regional aspects of methods and ways to study regional absorption, use of population PK, animal models, etc. And then finally also where we integrating these different tools, which could be either into complex in silico methods or in simpler decision trees or frameworks like a biopharmaceutical classification system.